Okay, this is Kei Sato from Institute of Medical Science, the University of Tokyo, Japan. And first of all, I'd like to thank the organizers for giving me the opportunity to present our study in this conference. So today I'm going to talk about our recent studies focusing on the evolution of SARS-CoV-2. So at first I'll briefly talk about the background. So maybe all of you know SARS-CoV-2 is the causative agent of COVID-19, and as of September 2022, almost 0.6 billion people were infected with SARS-CoV-2, and more than 6 million people were died of COVID-19. So meaning that SARS-CoV-2 infection and COVID-19 are ongoing pandemic in the world. And since the fall of 2020, we know that SARS-CoV-2 has highly diversified over 2 years. And at first we know that the alpha variant, it was known as B117, or the UK variant, has emerged in the UK. And then beta, gamma, and delta variant has emerged in 2021. And then all of you know, at the end of 2021, a new variant called Omicron has emerged in South Africa and then spread all over the world. And then to investigate and elucidate the characteristics of SARS-CoV-2 variant, and on January 2021, I have launched a consortium called the Genotype to Phenotype Japan, G2P Japan. And now more than 15 PIs in Japan, including in this consortium, to elucidate the characteristics of SARS-CoV-2 variant that emerge continuously. And since 2020, I have started focusing on SARS-CoV-2, and I have published 18 papers. And 12 of the 18 papers published, as shown in red in this slide. This study has been conducted by G2P Japan Consortium. And also one paper published at Nature Methods. In this paper, they focused on the methods for fighting emerging pathogens in the future. And in this paper, I have introduced the scientific activity of G2B Japan in this paper. Then in this paper, I have emphasized the importance of collaborative platform to combat emerging pathogens in the future. And also I emphasize to persist the fruitful collaborative activity to combat emerging pathogens in the future. So we know, based on the G2P Japan Consortium activity, we know that the scientific activity to elucidate the characteristics of newly emerging pathogens, in that time SARS-CoV-2, and then showing the information on the pathogens in real time is important. Therefore, we continue to elucidate the newly emerging SARS-CoV-2 variant now. And then, so today I'm going to talk about two SARS-CoV-2 variants called Omicron BA.5 and Omicron BA.275. And one paper focused on BA.5 has been recently accepted at CEL. So at first I talk about topic 1, focusing on Omicron BA.5. And this is a preprint focusing on BA.4 and 5, actually BA.5 now. And then this paper has been published at CEL now. So at first I briefly talk about the background on the evolutionary trait of SARS-CoV-2. So until the emergence of Omicron, we know that there are some variants of concern or variants of interest, which is called alpha, beta, gamma, delta, and Omicron. These 5 are variants of concern, and these 2, lambda and mu, are known as variant of interest. And as you know, based on this phylogenetic tree, this means that these variants are not phylogenetically related to each other. So meaning that in terms of the time on the emergence, alpha was the first one, and then beta gamma emerged in South Africa and Brazil, and then delta emerged in India, and then Omicron emerged in South Africa. But based on this phylogenetic tree, this shows that delta is not the progeny of alpha, and Omicron is not the progeny of delta, meaning that this variant emerged independently. But after the emergence of Omicron, as you can see in the phylogenetic tree on the right side, we know that the BA1 Omicron emerged at first, then BA2 emerged, and then BA4 and 5 emerged. And based on this phylogenetic tree, this means that BA2 is a sort of the progeny of BA1, and BA4 and BA5 are the progenies of BA2. So meaning that until the emergence of Omicron, showing on the left side, until the emergence of Omicron, it's kind of the antigenic shift-like evolution. But after the emergence of Omicron, the emergence of BA1, BA2, and BA4 and 5, these evolution are based on antigenic drift. And again, we can say that the evolution of Omicron, I mean the BA lineages, are posed by antigenic drift. So in this study, we focused on the characteristics of Omicron BA5, and we usually focus on 3 aspects of a SARS-CoV-2 variant in terms of transmissibility and immune resistance and pathogenicity. So at first we focus on the transmissibility of BA4 and 5 that emerged in South Africa in this year. And as you can see in this slide, we quantified the effective reproduction number of BA4 and 5 in South Africa. And then as you can see in this slide on the right panel, we found that the effective reproduction number of BA4 and 5 are almost 1.2-fold greater than that of BA2. And we usually focus on the mutation or substitutions in the spike protein. And when we focus on the spike proteins of BA4 and 5, we know that BA4 spike is identical to BA5. And as you can see in the slide, there are 4 additional mutations based on BA2. And then next we focus on the immune resistance of BA4 and 5. So for that, at first we used the human sera infected with BA1 and BA2 without vaccination. And as you can see in this slide, we performed neutralizing assay using these sera, and we found that BA1 and BA2 infection without vaccination do not induce efficient antiviral humoral immunity. But when we use the sera obtained from BA1 or BA2 breakthrough infection, I mean the breakthrough infection means the infection after 2-dose or 3-dose vaccination. And in the case of this sera, we found that BA1, BA2 breakthrough infections do induce antiviral immunity. But we found that BA4 and BA5 is almost 2-fold more resistant than BA2. And to address whether BA4 and 5 have the different antigenicity compared to BA2, we next use 2 kinds of sera obtained from rodents. One is the sera obtained from hamster sera infected with BA2, and the other is the murine sera obtained from BA2 spike RBD immunized mice. And as you can see here, we found that both of the cases in infected hamster sera and immunized murine sera, we found that BA4 and 5 is more resistant to BA2-induced immunity. So meaning that the immunogenicity of BA4 and 5 is different from that of BA2. Next, we focus on the pathogenesis of BA4 and 5. For that, we performed cell-based fusion assay. For that, we prepared two kinds of cells. One is expressing SARS-CoV-2 spike protein on the cell surface, which is shown in yellow in this slide. And the other is the human S2 expressed cells, which is shown in blue in this slide. And then we co-culture these cells, and then each cell expresses split respirase. So therefore, if these cells are fused, we can quantify respirase activity as shown in green cells in this slide. So based on this respirase activity, we can quantify the pathogenesis of the spike protein. Then in this study, we used the spike proteins of BA2, BA291, and BA211, and BA212.1, and BA4 and 5. And then this x-axis indicates the time after co-culture, and this y-axis indicates the pathogenesis. And this green dot line indicates the pathogenesis of BA2. So as you can see in this slide, we found that the pathogenesis of BA211 and BA4 and 5 spike protein is greater than that of BA2 spike. And then when we focus on the mutations in these BA2 subvariants and BA4 and 5, we found that a common mutation in BA11 and BA4 and 5, which shows a greater pathogenesis than BA2, beared a common mutation L452R. So meaning that BA4 and 5 and BA211 are more pathogenic than BA2 spike, and this greater pathogenicity is attributed by spike L452R mutation in this spike protein. And finally, to analyze the pathogenicity of BA4 and 5 and these BA2 subvariants, we prepared a chimeric recombinant SARS-CoV-2 based on the CPAR technique. And this chimeric virus, for the backbone of this chimeric virus BA2, and then we inserted the mutations detected in these BA2 subvariants as well as BA4 and 5. And we should note that the mutations in the non-spike region of BA4 and 5 are only 6 and 2 respectively. So meaning that although these viruses are still recombinant viruses, but we think that these would be almost similar to these authentic BA4 and 5. Then we prepared these chimeric viruses, and then we inoculated these viruses into respiratory human epithelial cells in vitro at first. And then in the case of airway epithelial cells, the growth kinetics of these variants are almost similar. But interestingly, in the case of human alveolar epithelial cells, we found that BA4 and 5 more efficiently replicated than BA2 in the lung epithelial cells. And then we inoculated these viruses into hamsters, and then we quantified the body weight and three respiratory parameters: pNH, RPF, and the level of oxygen in the blood. And when we particularly focused on the body weight, we found that the body weight of BA4 and 5 infected hamsters were significantly lower than that of BA2, suggesting that BA4 and 5 are highly pathogenic than BA2. So next we focus on the lungs of infected hamsters, and then at first we stained the nucleocapsid protein of SARS-CoV-2. Then at days 1, 3, and 5 post-infection. And then when we quantify the level of nucleoprotein positive cells at 3 days post-infection, we found that BA4 and 5 more efficiently spread in the lungs of infected hamsters than BA2. And next, we focus on the pathogenicity of BA4 and 5, and for that we performed H&E staining in the lungs of infected hamsters. And as you can see here, we found that the level of type 2 pneumocytes, which is the indicator for inflammation, we found that the percentage of type 2 pneumocytes at 5 days post-infection of BA5 infected hamsters was greater than that of BA2 infected hamsters. So meaning that BA4 and 5 exhibit higher pathogenicity and inflammation in the lungs of infected hamsters than BA2. And finally, now we have the infected hamsters with BA5. Then we collected the sera of BA4 and 5 infected hamsters and then used for neutralizing assay. And then as you can see here, in the case of BA4 and 5 infected hamster sera, we found that BA4 and 5 infection induces anti-BA4 and 5 immunity, exhibit anti-BA4 and 5 immunity, but we found that the humoral immunity induced by BA4 and 5 infection is not functional against the other variants, including BA2 and BA2 subvariants and ancestral SARS-CoV-2. So I will summarize at the first part. So in this study, we found that BA4 and 5 is more transmissible than BA2, and BA4 and 5 is more resistant to BA2 induced immunity. And we found that BA4 and 5 spike is more fusogenic than BA2, and BA4 and 5 more efficiently replicate in the lung cells than BA2. And finally, we showed that BA4 and 5 are more pathogenic in hamster than BA2. So now we can say that SARS-CoV-2 will evolve to increase its transmissibility in terms of the effective reproduction number. And also we can say that SARS-CoV-2 will evolve to evade antiviral immunity. And importantly, we can say that SARS-CoV-2 does not necessarily evolve to attenuate its intrinsic immunity, because BA4 and 5 are the protein of BA2, but its pathogenicity is greater than that of BA2, at least in hamsters. So I'll move on to the next topic. The topic 2 is focusing on the new variant called BA275. And here I show the phylogenetic tree of Omicrons, including BA1, BA2, BA4, BA5, and BA275. And based on this phylogenetic tree, we can say that BA5, BA4, and BA275, all of these are the progeny of BA2. But based on this phylogenetic tree, we can say that BA275 emerged independently of BA5. And there is only one common mutation between BA5 and BA275. That is R493Q. And also the place where the BA5 and BA275 emerged are different. BA5 emerged in South Africa and BA275 emerged in India. So again, we can say that BA2 is not the descendant of BA5. These two variants, BA5 and BA275, emerged independently. And then, because BA275 emerged in India, particularly in some states in India, so then we separated 10 states in India, and then we quantify the levels of BA275 and BA5 in each state and each month. And then now we can say that the Indian states where either BA5 or BA275 is spreading is different from each other. And particularly some states, they show that BA.5 is dominant, particularly in the south place of India. But in the north place of India, we found that BA.275 is started outcompeting BA.5. And then we quantify the effective reproduction number of BA.275 and BA.5 in each state in India. And then we summarize these values, and then we found that in India, the relative effective reproduction number of BA.275 is almost 1.2 greater than, actually 1.34-fold greater than that of BA.2, and importantly, the transmissibility of BA.275 is 1.13-fold greater than that of BA.5, meaning that BA.275 is the potential to outcompete BA.5. So next we focus on the immune resistance of BA.275. For that, in this study, we used two sorts of human sera. One is the vaccinated human sera, and the other is breakthrough infection sera with BA.1, BA.2. And then in the case of vaccine sera, we found that BA.5 is twofold more resistant to vaccine sera, and BA.5 is twofold more resistant to BA.1 to breakthrough infection sera than BA.2. But in the case of BA.275, we found that the sensitivity of BA.275 to vaccine sera and BA.1 to breakthrough infection sera are comparable to that of BA.2. So next, we used the hamster sera infected with BA.5. Now we are collecting the BA.5 breakthrough infection sera from humans, but for now we only have the hamster sera, so we now show the result of the use of hamster sera infected with BA.5. So now we found that BA.275 is strongly resistant to BA.5 induced immunity in hamsters. And then we focused on each mutation detected in BA.275. And then we found that some mutations, such as K457E and the others shown here, can contribute to the resistance to BA.5 induced immunity. So anyway, now I think we can say that the immunogenicity of BA.275 is different from that of BA.5. So next we focus on the affinity of the spike protein, actually BA.275 spike, RBD to humanized 2, using yeast surface display assay. And as you can see here, we found that the KD value of BA.275 was much lower than that of BA.2 and BA.5, so meaning that the binding affinity of BA.275 is much higher than that of BA.2 and BA.5. And now we can show that two mutations in the BA.275, particularly N450, N460K, and D339H, can contribute to the enhanced affinity to humanized 2. And then we performed the fusion assay based on the cells. Then we found that BA.275 and BA.5 spike are more fusogenic than BA.2 spike. Then we now have the authentic BA.2, Delta, BA.2, BA.5, and BA.275 viruses from patients. And then we prepared these viruses, and then we inoculated these viruses into human airway epithelial cells and human lung cells. And we found that BA.275 more efficiently replicated than BA.2 in lung epithelial cells, but not in tracheal airway epithelial cells. So suggesting that BA.275 prefer to replicate in lung of humans. And finally, to address the pathogenicity of BA275, we inoculated BA2, BA5, BA275, and Delta as a positive control into hamsters, and then we quantified body weight and 3 respiratory parameters. And although this slide is a bit busy, but when we focus on the body weight, we found that compared to BA2, the body weight of BA5 and BA275 infected hamsters were significantly lower. So suggesting that BA5 and BA275 are highly pathogenic than BA2, although these two variants are less pathogenic than Delta. And then next we focus on the lungs of infected hamsters, and then we stain the nucleoprotein of SARS-CoV-2 to see the viral spread in the lungs of infected hamsters. And then, as you can see in this slide, we found that the level of N-positive cells in the lungs of infected hamsters at 2 and 5 days post-infection. We found that the level of these cells of BA275 is significantly greater than that of BA2. So suggesting that BA275 more efficiently spread in the lungs of infected hamsters than BA2. And finally, we focus on the pathogenicity of BA275 in hamsters. And for that, we performed H&E staining, and we quantified the levels of type 2 pneumocytes in the lungs at day 5. And as you can see in this slide, we found that BA275 and BA5 exhibit higher pathogenicity and inflammation in the lungs than BA2. So let me summarize the second part of my talk. And in the study, we found that BA275 is more transmissible than BA2 and BA5. And also, importantly, we found that BA275 is more resistant to BA5-induced immunity, at least in hamsters. And also we found that BA275 spike is more fusogenic than BA2 spike, and BA275 is more efficiently replicated in the lung cells, human lung cells, than BA2 in vitro. And finally, we showed that the BA275 is more pathogenic in hamster models than BA2. So based on these findings, we think that BA275 will be the next risky variant of concern of SARS-CoV-2. And again, now we can say that based on the result of BA5 and BA275, now we can say that SARS-CoV-2 will evolve to increase its transmissibility and to evolve to evade antiviral immunity. But we should note that SARS-CoV-2 does not necessarily evolve to attenuate its intrinsic pathogenicity. So finally, let me introduce my lab, which is called systems virology. So as you know, there are many ways to focus on viruses, such as structural biology, cellular biology, immunology, pathology, and epidemiology. But now we want to understand, comprehensively understand the viral dynamics from microscale to macroscale. And I want to include all of these aspects into one study, and such kind of trial or such kind of study, we want to say such kind of study as systems virology. That is kind of the purpose or aim of my lab. So finally, and also we are now looking for new grad student and postdocs. So if you have an interest, so please just let me know. I'm happy to accept your offer. So finally, I'd like to thank all of my lab members shown here, and also I want to say that the result I showed today is not performed by my own lab. This is the study conducted by all of G2B Japan members shown here. So I just appreciate all of this effort on the evolution and on the elucidation of SARS-CoV-2 characteristics. So finally, thank you so much for your kind attention, and I'm not sure I have questions, but anyways, thank you so much for your kind attention. And if you have any interest in my lab, please just let me know via Twitter or email. Thank you so much.